Nucleotide excision repair, or NER, is one of the DNA damage repair pathways that eukaryotes have in order to fix large structural changes and helix distortions due to environmental damage. NER is commonly used to repair pyrimidine dimers caused by UV light and bulky adducts that bind to a particular nucleotide. When you spend hours outside, say at a beach, and forget your sunscreen, your skin is exposed to radiation from the sun in the form of UVA and UVB. The pyrimidines become covalently linked through cyclobutyl rings and pulled closer together. Additionally, smoking releases polyaromatic hydrocarbons, which is then metabolized and binds to guanine. Both cases cause DNA conformational changes and need to be repaired in order to avoid detrimental effects. There are two mechanisms of NER: global genome, which constantly scans the DNA for damage, and transcription coupled, which occurs during transcription when it stops at the site of damage. They differ in recognition of the thymine dimer. GG-NER. XPC, assisted by RAD23, recognizes the conformational change. XPA then binds to the pyrimidine dimer and helps recruit other proteins to the complex. TC-NER occurs during transcription, where RNA pull stalls when it runs into the damage in the template strand. CSA and CSB come to help recruit other proteins. XPA and CSA-CSB both recruit XPB and XPD helicase subunits of transcription factor 2H, which unwinds the 20 to 30 nucleotide stretch of DNA, including the damaged region. This is an ATP-dependent process. Replication protein A, or RPA, keeps DNA strands apart. XPF, ERCC1 heterodimer binds upstream of the dimer, and XPG binds downstream. XPF, ERCC1 cuts 15 to 24 nucleotides upstream of the lesion, and XPG cuts 2 to 8 nucleotides downstream of the lesion. This leads to displacement of the damage-containing oligonucleotide with transcription factor 2H, XPA, XPG, XPF, and ERCC1. The replicative gap repair proteins, replication factor C, or RFC, proliferating cell nuclear antigen, or pCNA, which is DNA clamp, and the DNA polymerase epsilon or delta bind to the 3' hydroxyl group generated by the XPF ERCC1 cut and carries out the new DNA synthesis that fills the gap. Once all the correct nucleotides are in place, the final nick is sealed by DNA ligase. And there you have it, a complete new strand of DNA that is fixed. If DNA damage does not get fixed prior to DNA replication, DNA damage is likely to lead to mutations.